Welcome everyone, and thanks for making the effort to come to the AGM. So this is, I think, the 6th AGM that I've attended. Is there anyone here from the first AGM in 2019? Yeah, that came along to that. Yeah, it was a very different time. But Race, or Recura, most likely, assuming that, has really evolved a long way in those 6 years. When I first joined Race, as an investor like you, I came in thinking there was something interesting about bisantrene. I thought the way the company had gone about trying to commercialise it was probably less than optimal, but I thought the actual drug itself actually had some potential. Little did I know how much potential it had. The continued evolution of Race Oncology, or Recura, is really quite an amazing transformation. I don't think there have been many good examples of a company changing so much yet staying the same. Considering that we're still working with bisantrene, still in AML, but we've added so much value through the application of science, effort, clinical. All of these activities have come together to create something pretty amazing. Effectively, with a biotech company, you're creating value out of nothing, and you're changing the lives of people. You're making hopefully a cure, or at least an improved treatment that will make a difference to many thousands, if not millions, of people around the world. 2025 has been particularly exciting year where lots of the activities have been going on for a long time internally, came public, and we were able to talk about them. First, obviously, composition of matter. This is something we've known for a long time, but in order to maximise the value of that, we've had to keep that confidential while we proceed through that. Second one was, this is something that I wish we'd discovered earlier, but unfortunately, sometimes it's just the way things go. Science is not a straightforward process. Occasionally, it takes a moment of inspiration and then a lot of hard work to turn that in. But the discovery around G4 binding, the G quadruplex mechanism of action, really transforms Race from, being another old chemo drug seller to something really pretty amazing. G4 quadruplex binding drugs are very difficult to make. There hasn't been a huge amount of success. People have wanted to make them, and all of the ones that are currently in development aren't particularly good. And we happen to have one that's really quite good at this and very effective. And then the identification of being able to silence MYC, being able to switch off MYC as a target, really has been, if you look through the literature, you ask any AI, the importance of this target and the value of a drug that can target this, you get ludicrous numbers. I think ChatGPT told me $700 billion is what the value of it is, which I don't know whether to believe it or not, but it's certainly confident. It doesn't matter, really. In reality, it's many, many, many billions of dollars. It's been the holy grail of the pharmaceutical industry to be able to target this gene and target this protein and be able to affect changes. As Pete knows all too well, this has been a very, very difficult target to go after for a whole bunch of reasons. And all of the attempts to date have not been particularly successful, and the drugs that have been developed have not been particularly effective. We actually have an effective drug that has the potential to be very, very valuable if we get everything right. And this has generated a whole bunch of new activities and building on and helping to explain why certain things have occurred in the past. One of the most frustrating things for me as a scientist is knowing that bisantrene does something but not knowing why. Why is it so effective? Why was it particularly? We did a lot of preclinical studies where we'd combine bisantrene with a whole range of different drugs, and it would all show synergy. Completely unrelated, had nothing in common with one another, and yet they'd all show this synergy. We now know why, and this has become really quite a valuable opportunity going forward. Lot to look forward to, and I'll start to take you through some of the areas. So looking first at the clinical pipeline, this has been something that's been evolving. The first two, AML and CPAX, or RAC001, mainly because I can't be bothered saying cardioprotection plus anti-cancer combination synergy. So CPAX, I think many of you are much the same. Both of those trials have been going on now for quite some time. If those of you can remember back to 2021, the AGM at that point, we announced these new programs in cardioprotection and in AML, and also reformulating bisantrene. If those remember, that were the three reasons that we raised the capital in the end of 2021. And those have all proceeded probably better than we may well have thought at the time. AML is now ready to go into phase 3, which is quite an amazing accomplishment for a company the size of Race. And the cardioprotection anti-cancer plus RC220 is progressing. It's obviously a difficult program, something that's never been really tried before. This idea of being able to better treat the cancer plus protect the patient from the damage caused by conventional chemotherapeutics like doxorubicin is a ambitious goal, but it's a difficult one to achieve. We don't have other clinical trials to draw upon, so we have to proceed cautiously. You can very easily kill a drug by rushing too quickly into trials. You have a bad patient have a reaction to the drug. The worst you can have is a patient die, and many drugs have died themselves along with the patient because people have got the clinical development wrong. So you really want to proceed cautiously through those things, but as we get through that process, things start to speed up. And then the new program, which we just announced a week ago, is Harness 1. This is looking at lung cancer, EGFR-mutated non-small cell lung cancer, particular subtype of lung cancer. I'll talk a little bit more about that in detail, in combination with a specific TKI called osimertinib. This is quite a large number of patients. You can see there almost 500,000 patients in China. There's an enormous number of patients in East Asia as well. And there's quite a lot of patients in Western countries. So it's not a small market. Currently, at least $10 billion a year of TKIs are sold, growing quite rapidly. And there's a real need for improvements in this particular indication. So this brings us to an area where MYC dysregulation is very important for a lot of cancers. 70% of cancers overall have MYC dysregulation, and it varies. Some it's quite low, 20%, to others it's almost as high as 100%. But these are 4 different MYC-driven pathways where MYC dysregulation is associated with resistance. These drugs that are used targeting these particular kinases, so either BTK, EGFR, KRAS, although it's not a kinase, it's a G protein, but and the MEK-BRAF kinase pathway. All of those are associated with a large number of cells, and they're all associated with resistance. All of these drugs work, if you have the right particular type of mutation, work really well. You can be almost completely free of cancer. You get given this drug, the side effects typically are fairly reasonable, certainly compared to chemotherapy or radiation. The cancer shrinks away to nothing. You're in remission. Everything's going really well. And then after some period of time later, 6 months, 12 months, 18 months, 2 years later, the cancer comes back. And at that point, it's resistant to these particular kinases. And this is a huge problem You have really good targeted agents, and every patient develops resistance to these particular drugs. Doing something about this has been one of the big unsolved problems in oncology, and we think we potentially have a drug that may be able to make some difference there. Obviously, this is a massive market. This is just an overview of one of these reports that you can pull off the internet where they estimate the size of the market. It's very difficult to get accurate numbers on all of these things, but it gives you an approximate idea of the scale of it. Right now, you're looking at around $55 billion just for these TKIs. That's an awful lot of sales for those. And all of those drugs, certainly the ones we've tested, have shown synergy between that and bisantrene, and potentially show, certainly for the EGFR, will show delay in resistance. This brings us to the clinical trials that we're proposing to undertake or are currently undertaking. And the first one I want to talk about is why we chose EGFR-mutated non-small cell lung cancer. So if we had the ability to pair with any particular TKI or targeted agent, why this particular one? And it was really a combination of clinical factors, preclinical factors, but mostly commercial factors. That's one of the great luxuries we have as a company, is we can focus on those areas where the commercial competition and the size of the market is such that we increase the probability of getting a commercial return for our shareholders. So we could go after some obscure, very rarely used kinase. Maybe it's in the public domain, the patent's already come to an end, a very small number of patients, that type of thing. Or we could go after something as large and commercially significant, where there's a great deal of competition between the various players in the market for domination. So at the moment, in EGFR, there's one major player, AstraZeneca, who sells Tagrisso or osimertinib, which is the generic name. They have about $7.5 billion as their expected sales for this year, all around the world, not only in the West, but also in China. They sell approximately $2 billion a year there, which is quite remarkable given the amount of competing drugs that are going on within China. It's kind of the gold standard for treatment for EGFR lung cancer if you have the right particular mutations. And it's very effective. The side effect profile is pretty benign, mostly skin type rashes and so forth. But the problem with it is that around 18 months, the median time, patients develop resistance to it and it stops working. And unfortunately, the prospects for most patients is pretty poor. 40% go on to no further treatment, so basically palliative care, and they have a very short life expectancy, a few months. Some go on to chemotherapy or radiation, and those patients then really don't. They get maybe 4 to 5 months out of it, and then they then progress. So there's not a lot of options at that point. Some of these other drugs coming through, developed by other manufacturers, there's 14 that are approved between Europe, US, and China, and there's no real clinical difference between any of them. First and second generation are inferior to the third generation. Third generation are all pretty much identical clinically to one another. They all have the same sort of side effects. They all have the same sort of efficacy rate. They all have the same time to treatment failure. The fourth generation coming through have proven to be pretty unimpressive in the clinic and haven't really worked out very well. And so there's certainly a great demand there for something that can improve the time, particularly the time to treatment failure. And you can think about it from a commercial perspective, it's really easy to put a valuation on that. If AstraZeneca is selling $7.5 billion a year of Tagrisso, and they get 18 months on average out of each patient, you can pretty much work out how much per month any increase, delaying the time to treatment failure, would add to their bottom line. And remember, of course, that's just purely profit, because all the cost pretty much is acquiring the patient, convincing the doctor to put them on treatment. Once they're on treatment, basically it's just the marginal cost of producing the drug, which is tiny in comparison to the price it's sold at. So you can start to see there that commercially this is worth a lot. The second factor that goes into that is the ability to take more of the market share. If you have a drug that's superior to everything else, there'll be a very quick change in the market dynamics. And that's what was seen with the first and second generation drugs. They had big sales. As soon as the third generation drugs came through, pretty much everyone, all the clinicians switched over to the third generation drugs. And the first generation now hardly sell any at all. There's a couple of specialised applications where they're used. So that's the second thing. So you have greater expansion of the market. And the third one is, of course, you get the value for selling your drug. So in this case, it's RC220. You get the value for selling RC220, and you can add that on to the values. You can start to see very quickly, you get very large numbers if you go through those calculations of what a drug like RC220 might be worth if we're able to demonstrate that we're able to delay treatment failure. And that's certainly the aim of what we're going to be trying to do over the next relatively short period of time. This brings us to the HARNESS trial, HARNESS 1. In lung cancer, for reasons that I don't know, you have to give your clinical trial a fancy name. I think it might be because there are so many lung cancer trials that it's not possible for clinicians to remember what the name of the trial is unless you give it. So we've called it HARNESS 1, which is Holt resistance in non-small cell lung cancer with EGFR sensitizing mutations. I think Marinella was responsible for this acronym. I can't actually remember off the top of my head now who came up with it. But the aim here is really to delay treatment failure. Ultimately, ideally, we would like to prevent it altogether. It's unlikely in most patients. Cancer is a very difficult thing to cure. But I expect in some patients it may well result in a cure. And that would be a fantastic outcome. We've certainly cured many mice of lung cancer. So unfortunately not mouse lung cancer, but human lung cancer. So it is possible to do, but we'll have to see what happens in the clinic. The aim here is to identify patients whose disease is progressing by ctDNA. So ctDNA stands for cancer tumour DNA. That is DNA that's leaked out of the tumour into the bloodstream, and you can by taking a blood sample, you can actually identify that DNA. There are particular techniques that are very, very sensitive, can detect down to a single molecule of the DNA, so that's as sensitive as you could possibly get, and identify whether that patient has a tumour and if that tumour is growing. And you can do that long before you can see the tumour by imaging, so be it by CAT scan or MRI. It has to get to a certain size, the tumour, before you can see it. But the ctDNA can detect those tumours when they're very, very small. So it gives us an early warning that the patient's about to fail treatment. So what we're going to do is look for patients that have ctDNA, try and make them ideally ctDNA negative, and associated with being ctDNA negative is a delay in the time it takes for treatment failure to occur. And also, it can also result in being actually cured from that. So in some trials that have been run in other diseases, if you can make a patient that's ctDNA positive and you make them ctDNA negative, remove all trace, it means you've actually completely eliminated the tumour and they're cured from that point onwards. That's the hope. We'll see how close we get along with that. So the endpoints, obviously, of this trial, safety is always the number one of any particular clinical trial. We want to work out what the best combination, the maximum tolerated combined dose is. Osimertinib has toxicity. RC220 at the high enough dose has toxicity. What we want to find out is what's the best combined dose. We have a pretty good idea what that will be, but we obviously have to go into patients to find that out. We want to look at changes in ctDNA. This gives us a really early indication of whether this is working or not. If we can take patients that are ctDNA positive after treatment with RC220, they become ctDNA negative, we've got a pretty good idea that we're on the right track. And we should have those results from very early on in the trial, maybe even from the earliest patients in the trial. They'll be running at the moment. We've got 5 sites in Sydney, Melbourne and Brisbane. We have the ability to add on more sites if needed. At the moment, that will be enough for us to accomplish everything we wish to accomplish within the cost structure that we have. Cost, around $11.5 million is our current estimate on the cost. We've done a lot of work. The team's been diligently working away throughout 2025, putting this trial together and getting all the costs and the likely costs. Of course, there will be slight variations, there always is, but that's approximately the cost, and that's before the R&D tax benefit. And because we're running this trial in Australia, we get back the full amount of the R&D tax, so any money we spend on this clinical trial. Time phase, we've got 9 to 12 months for the dose escalation. That's our best estimate at this point. That's assuming that, of course, we start and treat the first patient in Q1 of 2026. If there's any delay, of course, in starting that, because we don't have the funding, then, of course, it might take longer. The phase 1B will be faster, and that's because you can treat many more patients in parallel, even though there's more patients in the phase 1B stage. Of course, first patient is really what kicks all of this off. It's contingent on having the funding and having all the approvals in place. We've submitted the ethics application. At the end of September, we got the first round of queries back, responded to those, and they've gone back in to the ethics committee. And any day now, in fact, it could have even been today, I haven't spoken to the team, it could well come back and we could have ethics approval very soon. Most important thing from an investor's perspective, this is an open label trial. That means we can update you on progress. And certainly if we see anything really exciting like ctDNA responses, we'll be able to let you know. We don't have to wait until the end of the trial before we make an announcement. The frequent readouts are all of 2026 and 2027. And we expect to have the first efficacy data in 2026. So we should have some sort of idea whether this is working or not working, which is good. This is not taking years and years and years. I know there's been some chatter about this, that suddenly we won't see anything for the next 10 years. That's not the case. We'll get results very early on. This gives just a bit of an overview. It has really 2 sections. The ctDNA screening, so that's where we take patients that are on osimertinib or Tagrisso, their cancer's under control, they're being treated by their doctor, they're being watched just to see if there's any progression, but in the meantime, they're quite happily getting on with their life. We'll be taking blood samples from them every 6 weeks and testing that to see if there's any ctDNA present in their blood. And if there is, they then become eligible to come into either the phase 1A or the phase 1B, depending on where we are in the clinical trial and the interventional stage. And that's where we try and work out what is the best dose, and then also which is the most appropriate balance between. That's what 1B will do, is work out between, is going to the maximum tolerated dose the best way to go, or will a lower dose give less side effects and be just as effective or nearly as effective? And this is all what Project Optimus is all about. You have to nowadays take to the regulators, you have to be able to explain to them why you chose your dose level and be able to provide them with clinical evidence of that. You can't just say, I like the number 200, and that's the reason why the dose is 200 milligrams per metre squared. You've got to have clinical data. And so that's what the 1B will provide. And at the end of all of this process, we'll have an optimal RC220 dose in combination with osimertinib and have a pretty good idea if this is going to work or not. One of the great things about working with lung cancer, which is a terrible thing from the patient's perspective, is the disease moves very quickly once treatment fails. So the life expectancy of a patient at the point of osimertinib failure where the tumour is starting to grow is under 12 months, often less than 6 months. So you don't have to sit around and wait for a long time to know whether you get overall survival data, which is the gold standard. And as a consequence of this, many, many drugs have been approved very recently in lung cancer because of that at very early stage. So this brings us to this checklist. So I was talking about the possibility of accelerated approval. So the last 3 drugs in lung cancer have been approved either out of phase 2 data, it was about 200 patients in that data set. The second one out of 1B was 80 patients. And the final one was this sevabertinib, 70 patients. That was on Friday or Thursday. So the FDA realises you don't have to have a lot of data to give accelerated approval to a drug in this area. And the reason for this is the massive pressing clinical need for new treatments. They know that once the EGFR TKIs progress, there really is not many options for patients, and it's basically a death sentence. So what they really want to do is get these treatments to patients as quickly as possible, which is great for patients. It's also great for investors. Okay, you often have to wait many, many years to do late-stage trials, involve thousands of patients, hundreds of millions of dollars, 10 years. That is not ideal as an investor. You have to pay for all of that and wait for that time. Accelerated approval out of 70 patients, I think even our investors can wait that long. At least I hope they can. So what are some of the other things here that we went through? We went through this as we obviously wanted high clinical need. We wanted to have a proprietary companion drug, and that's certainly something that's really attractive. We wanted lots of people that we could talk to about this. So as I mentioned, there's 14 different TKIs approved already. There's 8 in phase 3. There's an enormous number in phase 2 and 1 coming in behind that. So it's a really competitive area. Any one of those drugs paired up with another drug that improved them could come to dominate the market. So you can think about it. If you're the dominant player, you want to protect your market. If you're one of the up-and-comers, you may want to acquire a drug like RC220, assuming, of course, it works, in order to capture that market. Obviously, we're building a lot of things around ctDNA, sort of the modern way. It'll only be a matter of time before drugs are approved on a proxy of ctDNA response, like they have with progression-free survival. Hasn't yet quite got there, but I would imagine within the next 5 years, there will be drugs approved on this basis. But for our purpose, we're not intending to get approval on ctDNA, but it provides us with a really early warning or an early signal of efficacy very quickly. And the final thing we really wanted to make sure that we had was we had a template trial process that we could apply to any particular TKI. The same problem that exists with EGFR lung cancer exists across the board in all targeted agent cancers. They all develop resistance. They all have ctDNA. They all have the same approach. So we can take the same design and move it across to different indications. And this is not just us doing this. This is anyone that acquires RC220 could also have the same idea. They can see how they can get what's called label expansion by doing the same basic trial over and over again. The masters of this are Merck with Keytruda. I think they ran 3,000 trials over the years for Keytruda, And they took the same basic idea and just applied it and applied it and applied it. And they ended up with a whole series of indications and a $25 billion a year drug. So the next trial that I'll talk about is the AML trial. It's quite a bit complicated because it's trying to do a whole bunch of things as efficiently as possible, as quickly as possible, in the lowest cost possible. Phase 3 trials typically are not very cheap to run. They take a long time. But we think that we've found a way of being able to accelerate and keep the costs down by focusing on initially a bridging study where we bridge RC220 based on the 110 data, where we show that they're identical. Many of you may remember me talking about how the PK and PD properties of RC220 were matched up to RC110. This wasn't just a random thing that we did. We actually put a lot of effort into ensuring that we got very similar properties. And the reason for this is to give us this ability of doing a bridging study later on down the track. So this has been possible for a long time and was built into the development of RC220. So in that case, you just have to compare RC110, you treat some patients, you measure the level of the drug in the blood, and make sure that they're very similar, very much similar to the way that generic manufacturers will show that their drug is similar to the brand name drug at the end of the patent process. Those studies typically involve, a dozen patients only. There's a few more patients in each of these, so 40 patients. And the reason is we also have to show under Project Optimus what is the optimal dose. And historically, bisantrine's been used sort of in AML in two different ways, and we have data for two different ways in the two phase 2 trials that we supported. One of them was over 7 days, and the other one was over 4 days. And so that's the intention there, is we'll compare 7 days versus 4 days. Obviously, dosing at a high level of RC220 for 7 days is going to be more toxic than for 4 days. And the question is that extra toxicity worth it for an efficacy point of view? And we'll hopefully have the answer to that. And so that gives us the data that we can take to a regulator like the FDA and say, okay, we've chosen 7 days for this reason, or we've chosen 4 days for this reason. So we have a meeting with the FDA. They then, in a discussion, you establish the optimal dose, and then you do a dose expansion. So you can take the same patients that were originally treated in the Part A, pull them across into the pool for analysis, and then you add enough patients on to get yourself to a statistical endpoint. Of course, the statistical endpoint is going to depend on a whole bunch of different factors and will be determined, but this is just my rough calculation of it, making a whole bunch of assumptions. A very cost-effective way of bringing this drug to market. You might think, well, why are we bothering doing this? Okay, there's a bunch of reasons. Number one, it gives us control over our own destiny. We can't sell a drug for EGFR lung cancer. That would require a sales force of thousands of sales staff, investment of hundreds of millions of dollars to try and do something like that. AML is a rare orphan disease, treated in a limited number of sites around the world. That is something that's within the capacity of an Australian company to do. So we have the ability to take this all the way through to commercialisation if we chose. So you've got control of your own destiny. It brings the drug to market as quickly as possible. This will get full approval at the end of this process for an orphan indication as quickly as possible. It'll be faster. Maybe if we get accelerated approval, you might get it onto the market and be able to sell it for a particular indication. But if you want full approval and all the various processes that go through that, you have to take it through a phase 3 trial. The other thing that this gives us is really valuable data around MYC. Unlike, if we want to look at is bisantrene RC220 able to affect MYC in a patient in their cancer, effectively almost in real time? That's very hard to do with solid tumours because you have to be able to take a biopsy of the tumour out of the patient shortly after they got treated. And that's very difficult to do with lung cancer. You have to put basically these really long, nasty needles into a patient, and it's a lot of risk. There's a lot of things that you don't want to hit with a big, long needle. But with AML, 90% of AML have dysregulated MYC, so it's a great cancer to use. The other thing is you can sample the cancer out of the blood. It's floating around, the AML's leukemia, floating around the bloodstream, so you can very quickly get the cancer out of the blood. And so we can get a dose-response curve all the way through the whole process very easily in patients from AML. So we will have, from the first patients in Part A, we'll be able to start to see whether we're having an effect on MYC in patients, which is incredibly valuable to have. So all of those things go together to say, given the cost, which is very modest, about $7 million for Part A, we get an awful lot of incredibly valuable data we couldn't get any other way. And this brings us to the current trial, the CPAX-1 trial, or RC010, you'll often hear it. It's our internal code. This trial's underway. As I mentioned, we're going slowly. We're slowly putting on more sites. Expect this to pick up in pace. We've been very cautious in how we've been going about this because not so much for RC220. In fact, we haven't seen any side effects from RC220 in the patients treated to date. But doxorubicin really is a nasty, nasty, nasty drug. And early on, we're not providing a huge amount of protection through RC220 because the doses are low. So they're pretty much fully exposed. We hope we're getting some protection, but not a huge amount. And so we have to proceed cautiously. Also, these patients in a dose escalation are very advanced patients that have failed all treatment, and they will have gone through a lot before they get to this trial. And so they don't necessarily have the physical capacity to be able to handle a big whack by a drug like doxorubicin if you're not careful. So it would be very easy to put a patient into this trial and kill them, not with RC220, but with doxorubicin. And if you were to do that, RC220 will get the blame for it because you won't be able to tell whether it contributed to the death. So you have to be cautious. Once we get going, though, I think pace will pick up, and particularly once we bring on South Korea. All the indications are that South Korea is the ideal location to treat patients. They're really keen to get going, and as soon as that happens, I'd expect the pace of treatment to pick right up. So slow start, hopefully quick pick up. Intellectual property, Pete mentioned this idea of being able to take a drug that was originally first synthesized in 1976 and be able to make it as though you first was made today. It's pretty amazing. By just following up some small peaks on chromatograms, working out exactly what's going on, a lot of investigation. Many of you will have, I'm pretty sure everyone here sat through the presentation Mike and I gave, Pete gave a few months back. We were able to discover that, bisantrene is not as simple as just a simple old boring chemo molecule. It actually has some really interesting chemistry, which I'm not an expert on why it does all these different things, but it does provide us with the potential for composition of matter. Hopefully we'll hear about that in the not too distant future, how that's gone. It's kind of a bit of a funny one. It's there's no particular reason why it shouldn't be approved. It's not as though anyone else has discovered this. It's not as though it's not useful, and it's certainly not obvious. So they're the things you need to satisfy to have a patent. So there's no one else has done this before, no one else has thought of this before, and it actually has particular use because if you allow bisantrene to be exposed to light, it becomes inactive over time. So we're as confident as we can ever be about anything in patents that we will get there, but we'll have to wait and see how that goes. But if it does come through, it gives us 20 years composition of matter, and that's a really great foundation to build these sort of other trials on and markets, because there's no point us going out and proving that bisantrene's a great drug to use in lung cancer if it turns out that we have no protection over it, because there's nothing then to acquire through any, and we wouldn't be able to sell it without immediately facing generic competition. So there's a lot of activity to expect over the next 2 years. I won't go through all of these. You can look at them at your leisure. So there's a lot there that will happen. I suspect there'll be other things that aren't on that list will happen, and with a list that long, there'll be some things that might go slower than we might like. But there's enough things going on. Once you've got 3 clinical trials running in different areas, you've got a lot to look forward to. They're all open label, all able to report upon, and if we're able to do what we hope, then we'll have a lot to tell you guys about. So it's not going to be a matter of sitting around for the next couple of years wondering what Pete and Daniel and the rest of the team are up to. So what are the key highlights? Well, we're in phase 3 with a de-risked and clinically proven anti-cancer drug for the same indication the drug was originally approved for. There's always risk in biotech. Anything can happen. But about as low risk as you could possibly get is to get an approval for an indication the drug's already been approved for. So in this case, AML. So that's pretty amazing. We're solving a real health problem. So either the damage caused by chemotherapy, particularly the anthracyclines, and TKI resistance. Both of these are super serious clinical problems. There aren't other solutions to that are clinically acceptable. In the case of TKI resistance, there's basically nothing really. And even in the case of the permanent damage, there's nothing that adds to the anti-cancer activity out there. So we're doing something kind of unique. We're going after really major markets. So in the case of anthracyclines, $1.5 billion a year of those sold, translating into sort of 4 to 5 million patients a year worth of treatment. They're a very large market. And the TKI, EGFR TKI market's over $10 billion. Best estimate is around 10. Some estimates as high as 13. Certainly a very large market. It's not a tiddly little orphan indication. The ability to silence MYC and being able to go after the undruggable holy grail target that everyone's been chasing is very, very enticing. And it's something that may trigger interest much earlier than normally occurs in the pharmaceutical industry. There are examples with companies that have come out with good preclinical data, have been acquired for large amounts of money, purely on the basis that the preclinical data they have is so attractive that people want to get in right away. And I'm not saying this is what will happen with Race, but it is a possibility that a transaction or partnership could occur sooner than many people might think. It may not require clinical data. It may not require an extensive amount of clinical data before something happens. And that's purely because of the target that we're going after. People have been trying to target this. They recognise this. Everyone knows this is an absolutely amazing target to go after. And to have something that can target this is very, very attractive to many, many companies in the sector. And finally, composition of matter claims really allows us to capture all the value that we're creating by doing these clinical trials and all this preclinical studies. Without that, none of that value can really be captured. So if I look back on where we are today at this table compared to where we were in 2019, where we had a drug with no IP, we didn't know how it worked, it was seen as being a boring old chemo drug, and we were trying to sell, I won't say we, the company was trying to sell as under the name patient program, versus where we are today. There really is nothing in common between those two things. Even the drug itself is not the same. You know, we talk about bisantrene, but now we're talking about bisantrene, not just this generic bisantrene. So everything's fundamentally changed. The only person left is Peter Websey from those days, the company secretary. Everyone else has changed, all the staff, the board, the strategy, everything. And that's part of the reason why we thought, you know, this is the opportune time. We're about to embark on a big step forward for bisantrene in the company, and to look at, you know, maybe Race Oncology has associations that really aren't relevant anymore, and we really need people to say, actually, this is something I need to look at again. I might have dismissed it in the past, but today this is actually a real company. Final takeaways, and this is some of the things here. There's really been no change to the clinical program since 2021. We're doing exactly what we set out to do in 2021, which was cardiac protection, anti-cancer. Remember that in those days, all we had was, you know, cell and animal data. We hadn't yet gone into the clinic. We had AML still on the program as we've continued to take that forward. And the third thing that we said in 2021 was to develop RC220. We said reformulate the drug, and we've managed to accomplish that. So all we've done is add some highly valuable additions in the EGFR through understanding the mechanism of action of the drug. So CPAX and AML remain clear priorities for us. We think there's very large value, particularly CPAX has got a lot of value. AML gives us a lot of optionality and really important data, so it's worthwhile continuing. So we've stayed on track. Of course, as we all know, there were some detours along the way, but you know, the vision has remained constant. Timeline commercial outcome has not increased, and this is something that I think was a misunderstanding by some people when they saw the strategy update, program update last week. They thought, oh, 10 years, they've changed direction again, it's going to be 10 years before we get there. And I hope you can see after this that actually that's not the case. We've actually accelerated and made many more opportunities for an earlier transaction to occur. And even if nothing comes of lung cancer or anything like that, CPAX and AML are continuing on as before. So nothing's changed in that side, and we've increased the opportunities we have to commercialise. The Harness 1 trial requires $11.5 million to start or complete. We have $5 million already raised thanks to people exercising their options early, and I'm very grateful for that. It makes a huge difference. We don't have a long way to go to get there. Many of you know that I have $3 million of that to go. I'm not going to sit around and see the trial not progress because of me. But we'll talk about that maybe another time. The most important thing to take away of all of this is these new trials will be run. It's not a matter of, you know, can we afford it? We have the funding. We can fund everything that we've announced out of the 125 options. You know, short of World War III breaking out or going into the Great Depression, it's pretty likely that those $1.25 options will be in the money. And if they're not, we've got bigger problems than anything else. So company will continue along. We won't start those trials, we'll delay them, but they will run at some point in the future. So, you know, why do we want people to convert the options? It's really because it's the cheapest way for us. It requires the least amount of dilution for shareholders, for people, because they're already there. They're already going to all get converted, so they're already built into the fully diluted expectation. If we go out and raise additional capital, that's certainly an option. There's lots of different ways that can be done. Some of it dilutive, some of it undilutive. But we will only do those things that make the most sense. So most sense would be for everyone to convert their options early. That's the best option. Second best is there's a whole bunch of them. There's dozens and dozens of them. Thank you very much.
Brian Walker, I am a shareholder. Thank you, first of all, for the great presentation and to the board, and particularly the staff and of the company have obviously done a fantastic job here. So if you guys are sitting in the background, I'd like to pass on my appreciation for a top job. My question is about the funding. Can I ask about the directors' options that are due at the end of this week? If they are in fact converted, the 265 options, would that not get us over the line for the funding?
Maybe because I'm the one who owns them, or at least half of them. I think it's not well understood is there's a lot of restrictions on those, the way that they were issued. They were issued under the 2018 Employee Incentive Plan. They have a valuation look back of 20-day VWAP, which makes it difficult to convert them. Second thing, so that means because the way the share price has been over the last month makes that difficult. The second one is they're not able to be transferred except under exceptional circumstances and exceptional hardship. So I'd have to be able to prove that I'm about to go bankrupt before I can be able to transfer them to anyone else. If I was to someone was to give me the money and I was to convert them and sell them to them, then I would then have a huge tax liability on that, which I then have to pay for. So in practice, they don't become a great way of doing that. If there's someone who wants to purchase a block, they can approach us at any time and we can do a placement for that block. You can do, what are called, what are they? Not a private placement. Yeah, well, just a placement. So outside of a capital raise, where you just do a placement to a particular investor at a particular price, and that can be done at any time. We have the capacity. We haven't done any capital raises for quite a long time, and so we have the capacity to do that. So that would be a better way of converting those. So the best way would be to let those options either convert cashlessly or expire worthlessly. That would reduce the number of shares on issue, and then sell equivalent number of shares to somebody who wishes to buy a block of stock. And that's certainly one way you could go about raising capital. So there's lots and lots and lots of different options that could be considered to raise capital if needed. But we may not need it. We've already had 5 million as of last week converted. We only got to get to 11.5. Remember, 3's still from me. So there's plenty of ability to get there in the time frame. So I think we should look and see, will the $1.25 options come in? And if they do, then there's no need to issue more shares and induce more dilution. But if it doesn't, well, then there's options to do other things as well, which will be, best for the shareholders. Ultimately, we've always taken this approach of what is in the best interest of the shareholders. Obviously, not raising any capital and doing nothing is not in the interest of shareholders. But at the same time, we don't want to issue lots and lots of shares and then have to do a consolidation and the like. I think all of us have invested in companies that have done that. You hopefully only once, and you learned. But you've got to balance those two things against each other.
Yeah, I just want to add as well, as the master of the budget process at Race, the piggyback options really do get us through these programs. We did approve the budget on the RACO20 trial to the end of the year, which is really about setting the trial up. getting the protocols written, getting the site set up. So the way I'm looking at the rate of conversion is we're highly likely to be able to initiate the trial without delaying it in any way, shape or form. The additional AML trial, the phase 3 component, that will really be cutting in after May, any major expense. So I think we're fine without, I've certainly never factored in the money coming from the conversion of those options. It's not in the budget. It's not assumed. So I think just want to stress that we do potentially, barring terrible misfortune, have the resources to complete all of the work that we've talked about today. So.
Hi, Madhav Shivpuri, shareholder. My question is regarding the Harness 1 trial. So given that, like lung cancer and the TKI resistance market is quite huge, as you have told us, just sort of curiosity, could that have been built into an endpoint within the CPAX trial itself, just to assess that? Thank you.
You mean ctDNA? Yeah, so we are measuring nucleic acids. I don't know whether anyone remembers. We said a whole range of biomarkers, and one of them we were looking at M6A RNA. Okay, so if you can get RNA, in the process of getting RNA, you will get DNA. If you get DNA, you've got ctDNA. So the answer is yes, but we haven't spoken about any of these sorts of things. So it's more complicated, though, with that indication.
Thanks for the question. My name's Simon Fisher. I'm the VP of Medical. I just wanted to add to the answer. So the 020 study, the study in lung cancer, is looking at RC220 in combination with osimertinib. The 010 study is looking at RC220 in combination with doxorubicin. So they're very different studies. They're combinations of different drugs, and they've got different endpoints. Daniel's absolutely correct that we're looking at some similar assays, some similar biomarkers, but in effect, no, we couldn't achieve what we wanted to achieve by redesigning or adding on to 010. Thank you.
Alan Saran, shareholder. First of all, congratulations to the whole team on getting where we are today. I'm astounded at the change over the six years or 5 years I've been holding. And when Daniel said the race is an iceberg, he was absolutely correct, but I couldn't have...
We're now allowed to talk about icebergs, are we?
I couldn't have...
Oh, we were banned from talking about icebergs.
Well...
If you're the Titanic, definitely.
So the iceberg was not a myth. My question is it possible that a TKI drug will be developed which does not develop resistance?
The answer to that is no, because of the nature of the way resistance develops. So it develops in two different ways. The first way is what's called bypass. So a TKI will be targeting a particular kinase growth pathway, and that pathway can be, there are multiple parallel pathways that can occur in cancer. And so if you have a driver pathway, let's say EGFR, that can be complemented. You can block that completely, and then another pathway, say MET, which is another kinase, it can mutate and take over the signaling to the cells. So what happens is you convert an EGFR-driven lung cancer to a MET-driven lung cancer. So that can occur with any TKI. So there's no universal TKI. Well, there are, but they're lethal, so you can't use them. You have to have something specific. So that's the first problem that you can't solve, bypass. And the only way you can solve that is pretty much targeting MYC at the bottom of that pathway. The second one is that there's a lot of diversity within a tumour. It's not just one, unfortunately, and this has been really, as we've learned more and more about how cancer works, you have multiple different subclones within the tumour driven by different driver mutations. So most of the cancer might be EGFR, but there's some few cells that are being driven by other drivers of that cancer, and they then tend to, if you block EGFR, those other ones are growing unconstrained because there's nothing blocking them from growing. And what happens is over time, they then come to take over. And so as a consequence, resistance is inevitable with all TKIs and also all targeted agents, basically, if you long enough. If you don't get to a cure, if you can't kill every single cancer cell within the patient, eventually the cancer will come back. And the targeted agents typically can't get that patient to kill every single cancer cell within the patient. So as a consequence of that, this is an inherent problem that's just part of the way targeted agents like kinase inhibitors work. And so you have to try a different approach to overcome that. And we think bisantrene happens to have the right properties that might allow us to do that. And that's what we're hoping to show in the clinic.
I'd just like to stress, again, Daniel mentioned it, that I was on the board of a company called Micarex for 7 years and stepped off that board because of the emerging conflict of interest with Race. So you can tell which horse I backed. And I think this centrality of MYC to these resistance processes is very, very important. And I want to stress that we're kind of on our own out there. There are a couple of other G quadruplex binders that aren't very, very good. And as I think we've discussed in the webinars, what makes bisantrene particularly good is that it's very, very insoluble. And so it's really difficult to get a drug like bisantrene through screening and development. It's just very, very challenging. I was talking to a medicinal chemist the other day and showed him the structure, and he laughed at it, because it's just, he said that's going to be insoluble just by looking at it. I don't know how they do that, but very impressive. And so that, being in that unique position of having a potent MYC silencing drug, and we believe it will silence other important proteins, that puts us in a fantastic position. And I think from a business development point of view, if in time, and this is some of the news flow that we will be generating in, I think it'll be over a couple of years, not waiting 2 years, but a process of exploring the different drug classes in combination with this MYC silencing activity. And I think we just start to see the potential universe that could be handled clinically just expanding and expanding. And there are a lot of big pharma companies out there that have numerous different targeted agents, targeting different proteins. And if we can demonstrate or start giving them the sense that, hey, this could actually prolong the time to resistance for that drug class, this drug class, the other drug, etc. I think it will be seen that we have something incredibly valuable. We did have the example of Mirati. Mirati was the first company to successfully target the KRAS G12C mutation. And, a couple of points from that case study. The first is they went from starting their phase 1/2 trial to a $12 billion market cap, even though they were only able to hit 15% of one target's mutations. They can't apply it to EGFR or other. It was just KRAS with a G converted to a C at position 12. They got up there. They got overhauled by Amgen, and so they did lose a lot of value. Amgen just could put more resources to it. So their market cap did come down. They were acquired by BMS for $4.8 billion with a $1 billion contingent value right. And the other thing I'd point out in that case study is they went from first in man with their drug to approval in about 4 years. So again, testament to the speed that you can move if you're working on something unique that's addressing a significant unmet need. So I just really wanted to throw that in and just point out that, theoretically, we have a bigger opportunity than Mirati ever had in its portfolio. And, the next period of time is about us exploring this and sharing that data with the market. And I'm absolutely convinced that that will continue to drive increasing interest, be it from investors, pharma, and obviously patients as well, who are very important. So, I really do think that we've got something extremely important in our hands. There will be other MYC inhibitors in time. That's inevitable. KRAS was considered to be undruggable. Now there are over 100 drugs in development. But as I said, I think the uniqueness of our position at the moment is potentially very valuable.
Ash Depler, shareholder. Firstly, thank you for your efforts over the last 12 months. It's been exciting to see everything come through. Question just around the recruitment for patients. Obviously, it's been going quite slow. You've got 3 patients there. You had 15 as the number that you needed. Can you talk a little bit about what's causing that hold up with recruitment? Is it a case of we need to market it better? Is it people are too healthy? What's happening there with regards to being able to recruit?
It's a matter of being very careful about which patients we bring onto the trial. We've screened a lot of patients. A lot of patients have been screened, they're just too ill to risk at this early stage. So there's no real solution around this problem except to add more sites, which is exactly what we're doing. So we've just recently brought on Hong Kong, 2 sites in Hong Kong. But the real thing that will speed up things is bringing on South Korea, and for a couple of reasons. One is they've actually got access to many more patients than any of these sites elsewhere, but also they're more likely to treat patients more early. The type of patients they've got access to, we can't get access to here in Australia. It's just the size of the population and the way the medical system works between these different countries. It's quite different, but that means we can get healthier, younger patients that are more likely to be able to tolerate the doxorubicin toxicity. So it's happening. It's not as fast as I would like. We're doing absolutely everything in our power to speed things up, and we hope to have news on this front over the next little while. But there's nothing more we can do that we aren't already doing, and it remains a real focus of us to get this going. I want to see this happen. This goes back to cardioprotection. Was an idea that I had back before I even rang up Race Oncology. I thought, maybe there's a possibility this will work. So I really want to see it happen. And if it does, it really does work out, it's going to affect a lot of people, literally millions of people around the world. So, there was somebody on Hot Copper the other day talking about their experience of Hodgkin's lymphoma, how it wiped out their cardiovascular, and they were a very fit person. So either they just made it all up just to impress all of us, but it really, rang true exactly the sorts of problems that are seen. These drugs work, they cure people, but they cause a lot of damage in the process. And if we could do something about that, it'd be fantastic.
Mason Hill, shareholder. Daniel, this is probably going to be a lot of questions that you can't answer. I'd just like to say thank you to the board and the team. It's been fantastic to watch this mature. The ctDNA marker, it seems quite novel. The time span from a positive ctDNA score to progression seems to be about 3 months. I'm curious, does the FDA recognise this time period, and that running this clinical trial, seeing progression be at 6 months, that would be 100% improvement?
Yes, ctDNA is not yet recognised as a provable endpoint. Certainly recognised by the regulators, and they're taking a lot of interest in it. They're putting out a lot of position papers on it. I think they issued guidance just recently, the FDA. There was a phase 3 in bladder cancer at ESMO, which I was at, presenting the cardioprotection mechanism of action. It was a big deal. Our first phase 3 trial where they were able to show a difference by using ctDNA as a predictive biomarker for the outcome. So they were saying if patients were ctDNA negative, they gave them a higher level of treatment, and they got a better outcome as a consequence of that, and they were able to ease off on the treatment. If they were ctDNA negative, they're less likely to progress. So this is coming. My guess is before the end of the decade, we will see drugs approved on ctDNA effects. But as a guide to whether you're likely to see an effect or not out of any treatment, ctDNA today, everyone recognises as ctDNA, a response, a change in ctDNA, either levels or status, is something that means that something's actually happening that's real within the patient. So you know your treatment's working if you see changes, or not working if you see increases in ctDNA levels. So all of this is coming, but we're kind of, I wouldn't say lucky, because it's part of the process of choosing EGFR lung cancer is the time from progression to one of the more conventional endpoints like PFS or OS, which is overall survival, is very short. So whilst we'll get a signal from ctDNA first, pretty quickly we'll have a PFS signal, which is an approvable endpoint, proxy endpoint, and after that, a very relatively short period after that, we'll have OS, overall survival data, and that's the gold standard. There's no query about that at all. If you're able to improve overall survival, then you know you've got a drug that actually works and is approvable. So that's one of the huge advantages about going after this particular indication.
Yeah, it definitely seems like an advantage. So the FDA recognised that this positive screening of ctDNA, that time span from that positive screening to progression, that is known by the FDA, and that seeing an improvement beyond that, say 6 months.
Yeah, well, that creates very large effect size.
Exactly.
If you're measuring something and it takes 3 months to get to an endpoint, like PFS, and then you change that from 3 months, which is just osimertinib alone, for example, to say 6 months or 9 months, that's a massive effect size. So you've doubled or tripled the expected time to resistance. And so that has two effects. One is it's very clear. You really don't even need to do statistics to see that, but it just means you just don't need very many patients. And it also takes away this question about, this often happens in oncology, where is 2 months really worth it, or is 3 months really worth it, or a month really worth it to a patient, particularly if it comes with side effects. This becomes a very clear outcome, so that there's no doubt that this is clinically relevant, and that payers will pay, and regulators will approve, and so on. So all of those things went into the thinking about choosing an indication, choosing a trial design that would generate the type of data that we would really want to see. So a lot of thought has gone into this. It wasn't just Pete woke up one day and said, Daniel, EGFR lung cancer, I had a dream, this is the way to go. This has been going on, and we've been thinking about this for a long time, debating all the possible options and settling on EGFR lung cancer as being the best opportunity. It's also, there are other things, not only is there MYC, but there's also other properties about G quadruplex binding that are really important to EGFR lung cancer or osimertinib resistance in this, and EE, bisantrene, has those properties that you need. So it's more than just a one-trick pony. So it's not all just relying on MYC. It's also on other targets.
Yeah, I'd just like to add that so Simon and I caught up with the lead investigator on the Harness-1 trial, and it was abundantly clear that I think the more innovative elements, like selecting patients using ctDNA, really attracted them in. It was an MD PhD, done a lot of work on minimal residual disease and ctDNA, and we had a fantastic conversation. Very engaged, but very much involved because of these more innovative elements that we're building into the trial. So it was a great catch up.
It's actually quite impressive. We basically have a drug that can deal with multiple secondary subclones through these different resistance pathways. You've identified a large market, but within that market, you've identified a patient population that is the most severely in need, where we don't have to wait a very long time to see a positive outcome. I haven't read about much into this, but it seems like ctDNA is kind of a simple molecular biomarker that connects with resistance and mutation across a wide range of cancers, a wide range of drugs. I'm assuming the information that we get from this trial will be informative to perhaps other things that you might explore.
Yeah, and I think the other facet to it is it does give us an early readout. If you're changing the trajectory of the ctDNA increase, then you know you're having an effect. And obviously we'd love it to go to zero in all patients. I don't think that's a realistic expectation, but it does enable this more regular monitoring, and as Daniel said, that will segue into firmer clinical endpoints. ASCO in particular this year, it was absolutely full of posters and presentations on ctDNA, the impact of ctDNA negativity on patients' prognosis. And some of the data is so bound or clear that I think it's inevitable that it will end up being used as an endpoint. And think about conditions like, for example, breast cancer. A lot of patients are cured of their breast cancer now, and so to do a trial for a new breast cancer drug, it's going to take years and years and years. And yet the correlation between ctDNA negativity and almost a flat line on the Kaplan-Meier curve, because patients are just simply not relapsing, whereas the other patients are, relapsing really quite quickly in some cases. It means that you can actually get, a new drug for a patient population. You don't have to wait 10 years to, unfortunately, count the bodies between the two arms to see whether your drug works. So it's going to be really revolutionary, because it will open up certain indications where, you just don't go because the trials are too big and too long. So it's definitely coming.
Fantastic. Thank you.
Samuel Gore, retail shareholder. I just had a question on the screening component of the Harness 1 trial with ctDNA, just getting clarification on what the minimum window can look like for a patient to drop in from being screened on their ctDNA sample.
So they have to be stable on osimertinib, which means they have to be, so the current inclusion criteria, they have to be on osimertinib for 6 months. So it means that they know that if osimertinib's not working for them, they would have relapsed long before then. They're able to handle all the side effects, so they're not intolerable, so that we know we've got a standard. So majority of patients can get to 6 months. So they're on 6 months, and then they're then screened every 6 weeks. Blood is taken every 6 weeks, and then they're looked for ctDNA changes over that time period. It means we have to screen many more patients than treat, but eventually all those patients that are on screening will eventually relapse. So just when they come into treatment could be the first blood sample that's taken, or it could be 2 years down the track. At some point, they will eventually progress and then go on. There is the odd rare patient that doesn't progress. I think one of the investigators we were speaking to said they had one patient that had been on osimertinib for 8 years, but it's very rare. If you look at most of the clinical trials, by 3 years, there's basically no patients left that haven't progressed.
Thank you.
Right now, the grandfather of Race, Peter Websey, has told me that the sandwiches are getting very dry out there, and we do know from experience, positive experience, that these Q&A sessions can go on for days left unchecked. So before I call the end of the meeting, I just actually want the Race people in the room just to stand up, and and I want to congratulate everybody for a fantastic job, well done this year. And with that, I'd like to close this part of the meeting. We can all move through. If you've got any other questions, we can definitely answer those. Is there one burning question over there?
My name's Michael McGeever. I'm a shareholder. Just wanted to echo the thanks and congratulations to the team and yourselves. I just wanted to understand what the kind of motivation for releasing the composition of matter patents now rather than when the trials were further progressed, especially with the new trials being announced, was. If you could explain that a bit more.
Yeah, it's always tricky. You want to leave it as late as possible, obviously, to maximise the reach of that 20 years of life. But there comes a point where you've got so many people under confidentiality, and there's that increasing risk that something is let slip, and that destroys your ability to get your patents and claims. So, nobody knows whether we could have left it another 5 years, but and the other thing is that just simply people start looking at a molecule more intently, when you're out there creating value and publishing data. So, yeah, it's a dark art getting it right. I think we left it pretty much as long as we could have done, but it was also that there were other patents as well that were affected by it, so we had to put that's why all three were filed on exactly the same day. It's so none of them could act as prior art for any of the other claims. So there was a lot of thought and strategy that went into that. But thank you for that question.
Yeah,
right.
It made a huge pain in the neck because you had to do 3 major patents simultaneously. So Mike will know what a scramble that was at the end to get all that together. So, but it puts us in a good situation where we're not just dependent on a single patent, single claim, and I think that's really important. Everyone worries, well, what happens if composition of matter falls over? It would change nothing, really. We would still have protection across the formulation, the manufacturing, the use of it. It's not possible to use it without protecting it from light these days, otherwise you'll inactivate it and you'll be putting a mixture of active and inactive drug into a patient which no regulator is going to allow. So all these different attributes around this all add to the protection. So we're not dependent on a single patent. We have a thicket of patents protecting the IP there. So, and that's really important to remember. So we're not just living by, or if the patent examiner decides they don't like a particular claim, the whole thing collapses like a house of cards. It's actually very, very robust.
Right, that's it. Let's go and have some sandwiches. Thank you.